Thanks for coming to the Penn Humanities Forum. I'm Jim English. I'm the director of the forum. This is the final event in the fall semester of our series on color. The series has been conceived and co-curated by my colleague, Professor Shu Yang of the English department, who's done a dazzling job and has my eternal gratitude for all the work she's put in to make this a consistently fascinating series of lectures and conversations ranging across the disciplines of the humanities and pushing against the boundaries of that term. The color program is going to continue next semester on January 21st in the International House with a brief 45-minute panel discussion featuring Bradford Young, a very talented young cinematographer, discussing the new black cinematography, and then there'll be a screening afterwards of his recent film, the film he recently photographed, Mother of George, which premiered last year at Sundance. So that's January 21st. We have that to look forward to after the holidays. But today our subject is beautiful science, the architecture of butterflies. This will be an opportunity to explore some astonishing convergences, or at least near convergences, of aesthetics and artful design with biology, environmentalism, and the physical and engineering sciences. To guide us into this nexus of beautiful scientific research, we have three distinguished members of Penn's faculty, Shu Yang, Daniel Janzen, and John Tresch. Shu Yang is a professor of materials science and engineering and of chemical and biomechanical engineering. I think there are also some kinds of engineering she's not a professor of, but I'm not sure what they are. She's a scholar inventor who holds more than 20 patents. In 2004, she was named one of the world's top 100 innovators under 35. She's been honored by the National Science Foundation and the National Academy of Engineering. In recent years, she and her lab have developed from their study of butterflies an innovative spray coating that duplicates the waterproof qualities of butterfly wings. Applications for this invention include spraying onto the surface of solar cells, if I understand correctly, to improve their efficiency. Professor Janzen is a professor and DiMaura term chair in the Penn biology department, an evolutionary ecologist, as well as a conservation biologist. He's one of the world's leading experts on tropical biodiversity. Among his many honors are a MacArthur Genius Award, the Kyoto Prize, and the Albert Einstein World Award in Science. He describes his work over the last 55 years as, quote, an exploration of the ecology of the interface between society and tropical wildland biodiversity. The ecosystem for his research is Costa Rica, and the specific site is the Guanacaste Wildlife Conservation Area in the northwest of that country. A great deal of his work is focused on the behavior of caterpillars, butterflies, and moths in Guanacaste, and I think it's safe to say that no one in the world knows as much about butterflies in that part of the world as Professor Janzen does. To frame the presentations of Professors Yang and Janzen and structure the subsequent conversation with them, we have John Tresch, Associate Professor and Graduate Chair in the History and Sociology of Science Department. Educated in Chicago, Paris, and Cambridge, England, Professor Tresch is a historian of scientific technologies whose highly original work draws on and extends the limits of philosophy, anthropology, and the sociology of knowledge. His book, The Romantic Machine, Utopian Science and Technology After Napoleon, published by the University of Chicago Press, won the Pfizer Award for the best book in the history of science last year. He's currently completing a book on the technical obsessions of Edgar Allan Poe, so it looks like even my own discipline is not safe from his voracious appetite for new knowledge. So I'll turn things over to John now to get us started. John?
Thank you all for being here, and thanks, Jim, for that warm introduction, and thanks to Dan and Shu and Qiming for organizing it. I'm looking forward to the talk today, and my job is just, as someone coming from the outside, to sort of set up the terms of the conversation, show some pretty pictures, and ask them the questions that hopefully some of you have, and then prepare the way for you to ask your own questions. Because I'm coming to this as very much an outsider to their fields, but I've learned in preparation some really fascinating things, both about their work independently and about their collaboration, which we'll share with you today. And I learned some things about butterflies. And that's why I was very happy to say yes to this invitation, because who doesn't like butterflies? They are wonderful creatures. They make you think of nice summer days, flitting through the air, a picnic. Their aesthetic qualities, the lovely atmosphere that they convey, their fleetingness, their ephemerality, all make them beautiful objects for everyone to think about. The fact that they also famously go through metamorphoses connects them to, I think, what's so appealing about them, the sense of their ephemerality, their sense of their short life, but also that they're always on their way between different points, that you think of them moving through the different stages of their development. They are creatures that even when they are pinned onto a wall, we think of as being involved in time, being involved in change, being involved in growth and development. And that's part of the resonance they've had for ages, for millennia, both in the sciences and in the arts. And that fluttering aspect of butterflies, the way that we think of them always in movement, is part of why I think they're also a really useful object for thinking of the connections between domains, between fields of knowledge, between the arts, between engineering and science. And that's part of what we're talking about today. And they have been the bridge for a number of people who connect science and art in various ways. Probably most famous is Vladimir Nabokov, who was not just an amateur lepidopterist, a student of butterflies, but actually discovered new species that he gave his name to, and came up with his own theories of the evolution and the origins of butterflies, which apparently, according to the New York Times a couple of years ago, are beginning to be taken seriously by some butterfly biologists. And this is the famous butterfly view of Nabokov with his net about to capture you. Some scenes from inside the Nabokov Museum, including the namesake species that he caught, and his famous butterfly net, which is also part of the mythology of butterflies, that they're there to be enjoyed by everyone, right? They're part of the ecosystem, but at the same time, we can all be part of the great pursuit of science in chasing them. All it takes is a net to capture them, and then also to collect them. So stamp collectors, coin collectors, add to that list butterfly collectors as just a way of being in touch with the natural world. Another more recent and a little more scandalous, well, I guess Nabokov is pretty scandalous in his own right, but equally scandalous artist, more recent, is Damien Hirst, who has put together, I can't call them paintings, but murals that are made out of butterflies in images that resemble stained glass in cathedrals, and have this kind of religious iconography to them as well. These are not paintings of butterflies, these are actual physical butterflies, and it was quite a controversial fact that 9,000 butterflies died to make his exhibit that he showed at the Tate. But that kind of gratuitous sacrifice of life in order to produce art is part of what Hirst is quite famous for, capturing life in frozen materials. In the lower left, his split open cows or split open sharks that are preserved in formaldehyde are how he made his name, with this ambiguity about, well, is this art or is this basically just taxidermy? Is it right to sacrifice animals simply for the interests of art? But of course, he's referencing all the other ways in which animals are legitimately sacrificed in everyday life, whether for eating or for the interests of science. And turning his attention to butterflies reminds us of the numbers of these specimens that are in drawers around the world. There's also a connection to another of his scandalous objects, which is this skull he made the skull itself is a natural skull, but he covered it with hundreds of diamonds, and it's valued at $98 million. So there's something that he's playing with here in all of these works about what are the materials of art, not just the symbolics of art, what does it represent, but what are the matters that you use to produce it? What are the different regimes of value that decide something can be used to be put on a shelf, to be put in a gallery, to be sacrificed ultimately, to be transformed, to undergo this metamorphosis that takes it out of ordinary use or other regimes of value and makes it into art. And of course, it's also wrestling with this idea that I think is central to butterflies, to our imagination of butterflies, which is passing of time, transformation, and ultimately death. Storing them, pinning them to a wall, putting them in a shelf is a way to keep them alive indefinitely, even though, of course, technically they're dead. As he said, butterflies give us hope because they look alive when they're dead. They're not rotting corpses, as he says, with a kind of gripping grisliness that leads to the scandalousness of his work. But this collecting and preserving of butterflies is part of the history of science, the history of natural history for quite a long time, and there are still vast collections that are hundreds of years old, a couple hundred years old, in all the major research universities in the world. There is something both fascinating, beautiful, and slightly grisly about this fact, right? That these creatures are all put in these orderly arrangements. But part of the fascination, I think, is that they are in some ways perfect scientific objects. Because as you've captured them, of course, there's always a narrative about capturing them. You have to go there into the wild, and you have a story about going to capture them. But as you've captured them, they preserve themselves extremely well, and they allow for this immediate visual kind of comparison. Lots of similarities and lots of small differences that allow you to reflect on what are the overall orders and ordering principles of nature. So for natural history in all of its moments, in its long history, butterflies have been great objects to think with. They're also connected to the history of preservation and recording in the history of science in general, not just because a daguerreotype here was taken of a butterfly collector. There's a kind of weird image inside the image, the frame inside the frame of this technology for keeping alive forever this man, which inside it has this other technology for keeping alive the butterflies forever, this kind of way of warding off mortality. But also because when the daguerreotype was introduced, this is the first form of photography that was widely available, daguerreotype. And it was introduced by an artist, Daguerre, who was a stage designer, who wanted to develop this technology as a way of more quickly painting opera sets and stage sets, and panorama sets as well. So it's a merging of art with science. He was able to get this technology patented by appealing to an astronomer in France, François Arago, who's also a physicist. And Arago arranged the deal to make it so that the daguerreotype would be patented and would be given Daguerre's name, and he would get a permanent salary for it, but also available to the whole world. So when he introduced the daguerreotype, Arago remarked that it's not yet the perfect technology because, for one thing, you can't take portraits with it because people move, and the way in which it developed at the time in 1839 that it was first introduced, it took a very long time and very bright sun for the image to develop. So you would just get these blurs if you tried to take a portrait of a person. But nevertheless, it was useful for still objects like fossils, which I think also have a connection to the specimens of butterflies. Another problem of the daguerreotype was that you couldn't make reproductions of it. You couldn't make lithography of it. You couldn't use it to print many, many copies. They're pretty much one-offs. an interesting thing about these images, too, is that they have the same kind of iridescence as what captures us in butterfly wings or in the neck of the peacock, right? There's a similar kind of shimmering quality that is not reproduced by the digital or the photographic reproduction of it. But the reason you couldn't make lithographies of them is that they'd be crushed if you put them in a press. But Arago said about this fact, the fact that you can't reproduce them. But would anyone imagine giving a strong pull on a band of lace or of scrubbing the wings of a butterfly? So there's no reason, he says. It would be a violation of the delicacy of this technology to try to mass produce it. And he does so by comparing it to what, in his mind, is the most delicate of human and natural productions, lace and butterflies. And what we're going to learn is, and what I've learned, talking to Shu and Dan, is that this idea that butterflies are incredibly delicate is false, that there is really rigorous architectonics to butterfly wings that are responsible both for their flying capacities and for the distinctive iridescence that goes with them. But I think it's interesting to think about the early technologies of capturing life, of storing things for eternity, being compared to butterflies and having this aspect of delicacy signaled. Now, that delicacy and changingness, that emphasis on metamorphosis, also has made them symbols of the essentially human in lots of ways. That which can always escapes being mechanized or made routine or frozen. I think if you've seen this film by Herzog, My Best Fiend, which is devoted to his friend and enemy, Klaus Kinski, you may remember this amazing several-minute-long scene where Kinski is playing with a butterfly on his hand. And for Herzog, this is a symbol of Kinski himself, the artist who is the actor, who's always capable of transforming himself, being himself a butterfly. That goes further back to the mythology of Psyche and Cupid, this Greek myth, where Psyche is associated with the butterfly, the descent she takes to the underworld, and then the eventual reunion she has with Cupid in the afterlife, where they're both crowned with immortality, are retold as an allegory of the soul's journey, the metamorphoses that we all undergo, entering birth and then entering the afterlife. And that's a long-standing mythology. So we've got butterflies in science. We've got them in technology. We've got them in the arts. There's also, from the early 19th century, a really fascinating way in which they're involved in society, too, in social theory. There's a social theorist, one of the first socialists, Charles Fourier, who was a utopian visionary from the early 19th century, dreamed up a system of phalansteries, which are organized social factories as well as living sites. Brook Farm, where Nathaniel Hawthorne spent some time, was modeled on a phalanstery. The idea was that civilization, as we currently experience it, limits us in all of our capacities. That we are many, many things, we have many, many passions, but we are forced into one narrow identity, one narrow set of occupations, one narrow set of things that we can do. So he theorized that the best thing to do would be to design a society where all the different passions of humans could be maximized and brought into contact and set in opposition to each other. And so he cataloged all of the different passions, those which are about the senses, those which lead to people coming together, forming different kinds of groups, but then also the passions that allow us to combine passions in different ways. So there's the Kabbalist, which it feeds that part of us that likes to have conspiracies. There's also the composite, which likes us to have harmony. But the crowning passion for him of all humans is here translated into English as the alternating, which is the passion to change passions, the passion to change our interests, to move from different activities, different interests, different people, different combinations of people. And that was, in French, the papillon, the butterfly tendency. So that which is most human in humans, according to Fourier, is the butterfly, this part of our soul which moves between things, which restlessly shifts from one passion to another. the anthropologist Edmund Leach criticized his fellow anthropologists in 1950 for studying different societies in a very bounded way and accused them of doing butterfly collecting. The funny thing is that in the 1950s, his awareness of what was going on in the fields of entomology, of ecology, of biology was extremely limited, because what was happening in the study of butterflies and other insects was much more interesting than that. And Dan Janzen is an inheritor of all the changes of the middle of the 20th century and has been a major contributor to changes in the study of butterflies, the study of insects, the study of ecosystems, and of conservation biology. He has a phenomenon of mutualism, which is always associated with his name, which is about the way in which ants and a certain tree evolve together to protect each other, that they are actually a single unit in a sense, that the ant has evolved to protect the tree, and the tree has evolved to provide food for the ant. So this interaction between two different species is necessary for the survival of both. It's miles beyond the butterfly collecting that Leach was criticizing. Likewise, looking at the way in which morphological aspects of butterflies are something you can see when they're in the drawer, but also are useful as adaptations to the environment, to deceive predators, to merge themselves, to camouflage, to merge themselves with their environment in different ways. And Jim, in his introduction, quoted Dan as talking about ecology as actually involving not just non-human species, but humans as well. And I'm hoping that he'll talk a little today about his conception of ecology as being not simply about creating nature preserves, but thinking about humans as also living creatures within the larger ecosystem that relies upon nature reserves, relies upon nature for not just biodiversity, but knowledge about resources crucial for humans too. So a much broader conception, not only of butterflies, but of ecology than was imagined in the middle of the 20th century. Shu's work equally takes off from, to pardon the pun, some of the morphological and structural features of butterflies. The work we're hopefully going to hear about today involves looking at the structural color of butterflies. The way in which the color, this iridescence that's there visibly in the butterfly, is produced in a very different way than a pigment or a dye is produced. So it's actually not, you cannot reproduce it simply by changing a tint. It's there in the actual molecular structure of the butterfly wing. That's where the color comes from. Likewise, to get back to Arago's point about delicacy, that structure brings with it various characteristics that are necessary for the butterfly to do what it does, such as hydrophobia. It keeps water off the wings of the butterfly, which enables it to fly. If water was caught on its wings, it would be bogged down, which of course can be very useful for our own engineering, right? For building buildings that resist water. What's striking in reading about Shu's work is the way, not just in the case of butterflies, which is the project that she and Dan are collaborating on, but in much of her work, she draws inspiration from natural systems and natural processes of growth to think about how to make materials that are useful in all the artificial and engineering settings that humans might want. So not just inspired by butterflies, but by giant clams, thinking about growing crystals as following processes akin to flowers. So just as Dan is blurring this line between ecology as being only about natural things and bringing humans into the broader picture of ecology, so too Shu is imagining engineering as being not inspired by and directly imitating the processes of nature, of growth of various kinds, of delicate, seemingly delicate processes, like those that form butterfly wings. So this is the conversation that they're going to have together, right? They're both breaking down their own boundaries in their own work, and they're breaking down the boundary between them. Usually, I mean, it's a great treat for us, for me, and I think for you too, to get to hear about butterflies from these experts. Because usually now, if we get to know them, it's by chance, because we happen to have one in our garden, or we see one on the street. Or deliberately, we get to go to a natural history museum where the recent trend is butterfly gardens, butterfly rooms, or butterfly pavilions where they're cultivated. So I think we're sort of in the role of these little kids getting to enter the butterfly pavilion with Dan and Shu. One thing I learned from an architecture student in preparing for this event is that the term pavilion itself is the English version of papillon. So because it was a tent structure, a light structure that resembles a butterfly. And so what we get to do in this meeting in the field, halfway between nature and culture, between art and science, is a meeting between these different fields in the pavilion that's formed here by the Penn Humanities Forum. We get to enter the papillon, we get to enter the butterfly. So I'm looking forward to hearing what Dan and Shu have to say.
So I'm Shu Yang from Material Science Engineering at Penn. And so what I want to tell you is a little bit how I actually started to look into the butterfly structures, how I interact with Dan. First, I want to show you this picture is the color in our daily life, right? So here shows a collection of different kinds of colors. And I know my kids will love this rainbow cake. But I want to ask you about this. Do you know where the color is coming from? Why there's different colors? Blue or green or purple or pink? Anybody has the answer for this? So for this one, for example, everybody knows about this, right? So if you it's depending on the soil. So if your soil is acidic or base, so they have different colors, so pH dependent. And this one, the soap bubble, when we blow the soap bubble, that's where the kids got excited, right? So actually, as you know, when you blow the soap bubble, it's a same membrane. And when light, especially if you're under the sunlight, they actually shine and generate iridescent and rainbow color. And likewise, if you like this, so there's butterfly color, there's the opal. Opal is actually, if you go to the Smithsonian Museum and pay attention about this, they said it's actually made of silicon particles. The silicon particle, they actually come together and generate the color. So all these things, the butterflies, or opals, the beetles, and more, and even for here, like why the sky is blue, it's all depending how they interact with light. It's all the structure of the surface. So as we know, the light, right? We said light is a spectrum of colors. So here shows, for example, you have a prism. You shine a light, and this one white light will actually bend and generate a spectrum of color. And so we said this is, in our term, we call light is a wave, right? And so depending on how they interact, so whether they bend it, so it's reflection, refraction, and the scattering, and depending how the light is traveling, that's why during the morning time we have blue sky, at night we have red sky. So the distance of the light traveling generate different kinds of spectrum of color, all right? And then if you make things more complicated and you're adding, for example, the membrane here is actually a layer of the structures. When light comes to this layers, depending, so this light coming here will reflect it on this layer, and the second layer will be reflected. If you have multi-layers, each light will be reflected, will interfere with each other. So it's a term, is they interact with each other and generate certain kind of color. And depending on the direction you look at them, and you have different kinds of color coming out. And also you can have so-called diffraction, you can have scattering, and so here shows a natural color. It's also called structural color. It's not because of the chemical reaction as we see in many of the flowers, leaves, and it's because of the pH dependence, or because of so-called pigments. The pigments is chemical reaction. And as we know, if you change the pH, you change color, or if you have anything like a paint, and over time they'll fade because the chemical reaction will be bleached. So all these things, it's because of the structure and light coming towards the structure, if the structure is not being changed, the color is always there. And it's all typically, as mentioned earlier, was John mentioned, and they always have some kind of shimmering, metallic, or shiny color. It's much brighter than typical pigments, all right? So here's one example. Oftentimes we show this as it's a morpho blue butterfly. And so here I want to show you two things, right? It's blue, very shiny blue color, and the color is actually very, very uniform. And so this is a very large area. And also, it's not so surprising, the butterfly wing actually is so-called water resistant. As you can imagine, if the water is actually soaking onto the wing, and they will get very heavy, they cannot fly, right? So if you look closer of the scale, this is actually how the butterfly wing look like. They have layers of this kind of scale structures. This actually shows, for example, normal scale looks blue. If you put alcohol, they become brownish color. People look at different kinds of structures of the butterfly wings. They have this different kinds of scale structures, and depending on different scale, and of course they generate different colors. And in the case of the morpho blue butterfly, it's very common. They have this kind of so-called lamellar structures. It's a layer structure, remember I just mentioned earlier, so that it's multi-layer reflection. So when light coming towards those scales, they will be reflected. There's many, many reflection generate those blue colors, all right? And so of course, there's a lot of question is, what is the origin of the color? And so this is just one example showing the morpho butterfly is the reflection, multi-layer reflection generate color. But even with the morpho blue butterfly, the structure, if it's based on the size typically is 200, 300 nanometers thick. Of course, in this range, there's a micron size, and based on the thickness, the periodicity, all right? So periodicity means repeating units, and you should generate blue color, but actually in morpho butterfly wings, there's in different species, they actually have different color. So it's a combination of the reflection, and scattering, diffraction, and also the pigments also absorb the light, and pigments are helping them to generate different colors as well. And other than that, there's many other kinds of butterfly wings. You may see this more from Dan's talk. So here, for example, it's so-called flicker, right? So when wing is flicking, they actually generate different colors. So when different species, or whether the bird or maybe a male or female butterfly or another species, another insect looking from different angle, when they flick, right, they see different color. So it's question of the shape. So here, the previous one I showed is perfectly lamellar structures. This is still lamellar, but it's tilted, all right? And likewise, this is another work. Actually, one of my collaborator, Alison Sweeney, when she was a graduate student at Duke, she was studying the butterfly, and here shows butterfly is actually polarized. They have response to polarization light. And they use this kind of, so depending on the light polarization, they actually have different colors, so you can see them differently. And so they use this mechanism for mating as a mating signal, all right? So why do we care, right? So of course, it's iridescent, it's pretty, it's interesting, and has all the structure we, as a material scientist or engineer, are interested in. And so, on the other hand, I actually have, at the time when we started to interest in the butterfly, all the structure color, we also started collaboration with another a very provocative architect, Jenny Saving. At that time, she was at Penn, a Penn architecture department. And so we started looking into, for example, can we use those for the buildings? Right? So here illustrate some kind of building features. So if you have this kind of modern building, this is actually from Nagoya, and you want to have certain transparency, right? But on the other hand, besides transparency, I also want to, for example, have some coloring, and in the facade, I want to have some aestheticity. And at the meantime, I want to have the building to be able to be keep the temperature, or keep it warm, keep it cool. And so there's a lot of questions of where the energy is coming from. So of course you have to use electricity here, right? You keep the room warm or keep the room cool. So the question is, can we put some kind of coating mimicking the butterfly? So either we can absorb the light, or either we can reflect the light and keeping the heat out. At the meantime, of course, I still want to read and work inside the building. I want to have certain kind of transparency. And also, as mentioned, for the building, one of the issue outside, if you want to keep the transparency, you want to have the building to be clean. So can we utilize some coating to repel any dust or self-cleaning? And also use them for water harvesting or noise cancellation and so on. All right, so we were actually, this is almost like slightly less than 10 years. When I first got to Penn, I was at Bell Laboratories, Lucent Technologies, as a staff scientist. We're working on so-called photonics. So it sounds like big name, right? But actually, you actually use this at a daily life. So photon means the light, right? So we work with photonics devices trying to guide the light. And one of the methods is to generate so-called photonic crystal. And this photonic crystal will be able to reflect the light. And then if the crystal, any light comes towards the crystal, is totally reflected, and this crystal is stopping light. But if you drill the hole, then light will go through any of the holes and guided light. So that's why, and when I was at the Lucent Technologies, we're interested in generating this kind of the wave guiding photonic crystals, and to replace, for example, your optical fibers. And one way to generate this photonic crystal is use so-called holographic lithography. So if I say hologram or holographic, right, you know this is something is light coming toward the surface will be reflected, interfered, generate the 1D. Typically, if you have two laser beams come together, you generate 1D structures, and you see some coloring if you move around the film. So here we actually put the 4 laser beams together, so we generate 3D patterns into a photoresist, and this photoresist is commonly used to make your chip. All right, so we generate the 3D structures, and it's typically micron size or sub-micron size, micrometer size. And so this is it's comparable to the wavelengths of the light, and that's why they generate the color. And of course, it's because of the light, how the light is coming. It's similar to the multi-layer reflection. You generate different kinds of iridescent color depending how the light is traveled. And we make this a little bit more sophisticated as we control the chemical reaction. We introduce a little bit of roughness on the surface. So this is, as you can see, it's very, very smooth. And this one a little bit rough. So it's mimicking the butterfly wings as well as the lotus leaf. So they repel the water. So it's not only it has color, but also is water repellent. All right. So all the color we show, it's great. It's iridescent, has rainbow color. Any angle you look at is pretty. So when it's pretty, of course, you can use for different kinds of arts, and you can use for the cosmetics. But there's one drawback, right? So which means if you look at from different angle, you have different color. So this may not be actually desirable if you have a screen. I want to read this book. I want to read on this computer. Right? Each angle I see is different color. So for display, you don't want to have this kind of iridescent color. But in nature, also they solve this. They use this, so in the morpho butterfly wings, this lamellar structure, the height is actually not exactly the same height. And so if they are not exactly the same height, so the color is actually sort of diffused, just like our projectors. It's somewhat, there's a diffuser, make the color more uniform. And likewise, in this bluebird and the beetles, they have so the structure, as you can see, it's not really, really ordered. It actually has so-called local ordering, so it looks like hexagonal, but long range, they don't have long range order. So it's a mix of crystal and amorphous structure, make them actually more uniform colored, all right? And so there's also, if you make them less uniform, become totally disordered, what you get is actually white color, right? So we see here, for example, in the milk we drink every day, the milk is actually made of particulates. All right, so any of the fats they generate, it's emulsion, and all your water emulsion, it's particulates, they keep bouncing around and reflecting light, scattering light. That's why it becomes generated white color. And this one is very interesting. It's discovered 2007, reported by 2007. And what the optical physicists discovered here is this one actually has, the scale has about 5 micron thickness. And this kind of random filaments is 200 to 300 nanometer, and make this very, very white. So here the white give you some, the whiteness is 60. So what does that mean, whiteness 60? So think about if it's unpolished paper, you get a whiteness about 65 to 70-ish or so. All right. So to get the white paper, you need to have millimeter size of this cellulose into the paper to get this whiteness. All right, and this one is only 5 micron, all right? And at the same time, so we reported in 2009, we were making this kind of nanostructures, and depending on the size of this, we generate different kinds of color, red or blue color, and my student accidentally put them into the water, and they generate this kind of structures. All right, it looks detrimental here, but actually they generate different kinds of color, which is also interesting. All right, and this is also of only a few micron size. So this draws us to think, can we switch them? Can we actually manipulate them? And depending on the geometry, the periodicity, the ordering, can we actually manipulate from the color to whiteness? All right. So you may ask why we care whiteness. All right. Of course, I said as a paper, it needs to be white to write something, the ink, the black words on top of it, and beautiful, the white flowers or cake, and teeth needs to be white. Everybody spend a lot of money to make the teeth white. But technologically, right? Technologically, so I'm sure a lot of people using the e-ink, the Kindles, and the iPad. So for this one, the early generation of the e-ink is using the pigments. They have white pigments and black pigments under the electric field switch, all right? And that's why you have the white and the gray and white and black. And actually, more longer history is actually all the display. Any of your computers, right? You need to have some kind of display, so you need to have actually a white reflector underneath. Anything on top of it will be reflected, so you can increase the contrast, which is very, very important to have high whiteness, and it's very expensive to make them. And this is another, I just saw this today in the newspaper talking about this, using the white roofing. And typically in the roofing, we use black roofing or sort of like brown, darker color roofing. But those will absorb the sunlight, all right? Of course, this may not be issue in Philadelphia, but if you live in a warmer place, and if you absorb too much of the sunlight, this is, well, getting very hot. So people started to put a white roofing trying to reduce the solar gain, all right? So this is just another example. And then after we published our paper in this butterfly mimic, right? So at that time, we are just engineers looking at the color, and we say, oh, looks like butterflies, so we generate pretty pictures, and to attract interest from our peers. And then, of course, because the color is pretty, so Penn decided to put this onto our Penn Today news. And then I got a lot of people inquire about this, and then I got an email from Dan. And then said, I actually work on butterflies. So we started to talk to each other, and I started to know more and more about the butterfly wings, and then said, well, there's many butterflies more interesting than muffled butterflies. And sometimes they have this kind of white spots or patterns, all right? So this is so-called eye spot. You probably hear more from Dan. And so then we started to, this is over a year now, right? We back and forth talking to each other, and then Dan introduced us to, he gave us basically very generously, give us probably 100 butterfly wings. And we have no idea how we deal with this. So we just randomly pick up some of them, and this is one of them, interesting, very interesting. And actually Dan brought us attention. He said, this is a Caristodes, right? So normally, if you in the field, you look at from straight on the vertical, so lower angle, you actually see the white spot. But if you're not careful, for example, as a museum staff, they put into the drawer, you don't see them. All right? So we decided to take a look of the SCM, the scanning microscopy images of why there's a whiteness. So they actually saw this random arrangement of the scale, and under each scale, of course, it's actually highly ordered. All right? And this is a fine structure of those order structures. So it's because they are random, and also they have some tilting angle. So depending on which angle you look at them, you either see them or you don't see them. All right? And also Dan told us there's also differences between the male and female. If you look at carefully, right? What is the difference between those two? Can you tell? Obviously, this one is different. Any other differences? I'm not sure that's true. That's probably just the photography. And if you pay attention here, right? So you see the spot. And this is, you see the spot. This is just taken, this is taken by my postdoc. This is taken by Dan. All right? And as you can see here, you see the spot in the female, but you don't see here. All right, so again, it's illustrating there's some kind of angle dependence. The male and female are different. All right, so again, coming back to us. So I want to wrap up on my talk. So why we care, right, as engineers. So there's many interests actually trying to understand how in the biological examples, they change color and change the transparency, whether we can utilize in technological world. Whether we can change the transparency, for example, we can change color, right? And so this brings back to the collaboration I had with Jenny Sabin, is to introduce some kind of coating of building skins or facade. They can change transparency depending on the light, whether they are transparent, when it's low sunlight, whether it's a lot of sunlight, we want to block them, all right? And so we start to work on different kinds of patterns. So this is one example by my student Elaine Lee. We introduced those kind of pillar structures onto the wrinkles. And so as you imagine, if the pillar is it's wrinkled, the pillar is tilted, it's random scattering, it's opaque. And when you're stretching this, when showing here stretching this, and you actually make the pillars stand up, so they're more or less colored, and keep stretching them, they become far away from each other, you don't see them anymore. So that's why they become transparent. So this brings us to think about this is whether we can design this, right? We use sort of opal structures, the silica particles, and we embed it into a polymer film, and you don't see them. So it's completely transparent. When you're stretching them, you started to have opaqueness, a little bit colored. And this one, I guess I have to manually activate this, and then we got smarter to design our material, and we started to put Penn logo, all right? Oh, this way. So the last piece I want to show is, well, we do this manually stretching, of course. When you put it into the building, you can't stretch the building, right? So we decided to work with our colleagues in the engineering, Jan van der Spiegel and Nader Genta. We designed together with Jenny Sabin. We put into those, this one I guess also needs to be activated. All right, so here shows we put those particles we designed in our labs and put into the typical display, so-called indium tin oxide the glasses. And so we introduced this silver dots are actually photo diodes. They can sense how those hands is moving towards them, changing the light intensity, receiving onto the board. And then we introduce, once light intensity is changed, then we introduce a voltage. It's actually 1 or 2 volts, very, very low voltage. And you can change the color depending how your hands is moving towards them, so they can actually sense the light intensity change or the motion, all right? So we can do this wirelessly as well. So there's a lot of excitement about this from our point of view, is how do we actually eventually is making them reels of these kind of materials and very cheaply. So that's why we want to continue to work with them and also work with other biologists, understanding the mechanism in nature and trying to understand the strategy, the converging strategy, and see whether there's any particular benefits or the particular advantages to do certain kinds of structures. So this morning we just submitted a proposal together. Our hope is to bring a center, a huge center, to Penn, so we can work with biologists as engineers and biologists and physicists and chemists all working together to actually explore more type of biological examples and trying to impact the human habitat. All right.
I am on campus half of the year, and in Costa Rica the other half of the year. And when I use the word I really mean two of us. Winnie Hallwax, my wife, is sitting down here in the middle, and we do all of this together. Now, Costa Rica is a very small country, as you can see on the bottom of this slide. Transposed it on the United States, you can see how it is big relative to the United States. Size of West Virginia. That's where my laboratory is. And my laboratory is this yellow piece up here in the corner, northwestern corner of Costa Rica. That's the size of Philadelphia and its suburbs, to give you some sense of scale. So it looks like on the ground, ocean out here, Pacific dry forest here, high cloud forest, cold, wet cloud forest on the tops of these mountains, and Caribbean rainforest over here on this side. Now, when I say my laboratory, what I really mean is these are my clients. The 375,000 species of things who live in that area the size of Philadelphia and its suburbs. And my goal as a conservation biologist is that they're forever. They survive as long as infinity. This is a client interview. These are farm children, 4th and 5th grade. Family income's $3,000 to $5,000 a year. Their teacher sends a photograph to me of one of them, because she's proud of him, handling a snake, a live snake, and showing and explaining it to the other kids. That's not what I see in the photograph. What I see are 5 cell phones taking photographs of it. Out of the pockets of very, very poor farm children. That's the generation I deal with. That's the populace that I deal with. Those are my students. But it's not just the kids. It's also the parents that go with the kids. And it's not just wealthy people, like the ones in the last photograph, but it's also people at the other end of the economic scale. And when I say we here, I'm referring to really a team of people. These are the people in my lab. And it says careers. Notice it doesn't say undergraduates, graduate students, or postdocs. These are people who spend their lives doing what we're talking about today. And I call them biodiversity managers, because what they're really doing is trying to understand and manage this big chunk of biodiversity. The formal name for them is parataxonomist. The word is borrowed from paramedic or paralegal. There's one university degree in that photograph. 2 people who've gone to the university in the photograph, and the rest of them maybe made it through grade school or high school. As an academic, an ivory tower occupant on this campus, what I do is find all the caterpillars who live in that place. So I really wear 2 hats. We work at conservation, and we work at basic science. And people say, why caterpillars? It's because I fell in love with caterpillars when I was 10 years old. I am being paid by the University of Pennsylvania to do what I was doing when I was 10 years old. Okay? Now, what does a parataxonomist do? Well, there's one climbing up a ladder to get a caterpillar that she saw the turds of on the ground. Next door, and there she is with the caterpillar right there. Goes in a bag, goes into a laboratory. Notice the elegant chairs that they work off of, where it's databased, computerized. It goes to a rearing barn. Each caterpillar is in one bag, and eventually produces an adult, which gets mounted on a board, dried in an oven. This is the same kind of work you pay $70,000 a year to a Smithsonian technician to do. And product looks like that. These are the butterflies that you've been seeing all the way along here, but now there's a lot more species in that box, and there's a lot more variety of color in that box. And what I'm going to try to do today is just touch on some of the reasons why they have those colors. Shu looks at how they have those colors. I'm intensely curious as to why they have those colors. Now, notice already, those are the products from the caterpillars. And the products from the parataxonomist, finding them, rearing them, and preparing them as museum specimens. So now let's look at why. Now, where do they go after that box? It's a box full right there. It's a box size of a typing sheet of paper. There's a stack of them there going across the street into the Smithsonian Institution. They go in large numbers like that, and they end up filling up cabinets in the Smithsonian. This is your tax dollars at work, paying the rental space for them on the most expensive real estate in the eastern United States. Okay? Inside those cabinets, they look like this. So far, this is ivory tower, collect butterflies, organize them, think about their names, get them stored, do all the stuff that hardcore scientists do, right? Well, this is a tabletop with some of those drawers out and open on it. And I walk up to that tabletop and I pick up the drawer. And now normally, and Shu referred to this, as we go back and forth to the cabinets, you hold the drawer like this. It's just out of instinct, flat. You don't do this with it. But that one day, as I walked through, I think there was a table there, and I had to bend it up or down. I can't remember which way. And the late sun was coming through the window. And as I turned it down like this, the drawer sparkled. And it caught my eye. I looked at it, wait a minute. And so I did back and forth, and there were little white spots that showed up when I tipped it sideways, but when I looked at it the way we as biologists always look at it, straight up, nothing. Well, that drawer was full of these butterflies, which is what Shu was showing you. That grayish looking area there, looked at flat on. You see, you're looking at it now at right angles like this, flat on. It's the way taxonomists and biologists in the museums always look at them, it's flat on like that. That photograph I took because I wanted a picture of the whole butterfly. I wasn't thinking about what we're talking about today at all. But I went back and found these photographs after these little white sparkles came out of the drawers. And I discovered in the same set of photographs this picture, which I had taken because I wanted a picture of the face. But look at the white spot. Suddenly that gray spot turns into a bicycle reflector. Let me go back to it there. See there? Nothing. Straight on, it looks like that. That's a signal. On, off, on, off, on, off. Now, there are many different reasons to have signals, but I happen to know that the males do this very well, and the females don't do it hardly at all. So that's part of the difference, how mom knows who dad is, okay? Or the boyfriend knows who the girlfriend is. And then when I began to put these things together and Shu appeared on the scene, of course my reaction was, oh, how does that happen? So what she does is take these really neat pictures, he in her lab does, that shows that in that area right there, the scales are standing on edge instead of flat, as they are on all the rest of the wing. So that when you look at it straight on, you're looking right down the edge of the sheet of paper. You don't see hardly anything. But if you look at it at an angle, you're looking at the side of the piece of paper and then you see the white. Okay? This causes one to start thinking about white. Because everybody studies those blue morphos. I'll show you some living ones in a moment. They're sort of passé, everybody looks at them, but white is everywhere in the biological world, and people don't think about that. So you start thinking about that white, and look at this. This is a very common butterfly in the United States. It's had a name since 1863. This is not rare or uncommon, okay? I caught them when I was a kid. Lots of people catch them. They live in your backyards in Texas, California, and Florida, and Pennsylvania, and so on. Well, we go out and get a bunch of them in our backyard in Costa Rica and look at their genetics and discover, oh my goodness, there's a second one that looks just like the other one flying around with it. Doesn't even have a name. So you sit and you stare at them, this one and this one, for a long time trying to, how do I tell them apart? All of a sudden the light bulb goes off. See the white spot right there? That's a white spot on the male scent patch. This one doesn't have it. I'm sure that's how the males and females tell each other apart. Okay. Look at these two flies. They are virtually identical. They look like the size of a housefly, and they're virtually identical as houseflies sitting on your tabletop. Look at the face. This one, white spot, white silvery spot there, a gold spot here. That's how those two things, these are two species now, how these two species know who is who. So when you see a photograph that looks like that, you say, oh, look, 6 species of butterflies. This, in fact, you say, oh, no, they all look alike. Start looking at the white band right here and right there, and the degree of white and the degree of white in each one of these things up here. And you begin to realize that when this thing is flying and the wings are going up and down and up and down and up and down like that, each one of those 6 species is showing a slightly different blur of black and white and blue. And that's how they tell each other. They all occur together. So how do you tell, I see this one, is that my female over there, or is that not my female? Is that my male, or is it not my male? Okay. Now white is everywhere, and it's not always pigments, or it's structural in some sense that we've been talking about. These are butterfly eggs. Well, you know how big a butterfly is. We can imagine how big the egg is. And then this is a wasp who's a parasite on the eggs. Well, these white eggs are not white now because, at least it seems as though, they're not white because that gives them some evolutionary advantage to be white. But rather, this is a white which is a straight serendipitous outcome of the structure that Shu was just talking about. You can see the ridges beginning to quite visibly down here. And if Shu takes one of these things and drills into it the way she does, it will have a very distinctive pattern. I don't know what it is, but it'll have a very distinctive architecture. Okay? Now here's the guy that everybody knows. It's on all the tourist brochures, and when you go on your eco-tourist trip, you'll see one, I'm sure. That one right over there, the morpho butterfly. That everybody knows about. That's what Shu was showing you the photographs of and so on. Well, let's focus first on this guy. These are the scientific papers written about, and this is a different species of morpho, but scientific papers written about them. Fine. Okay, let's go back and look at this. Now, the first white question I have is, of course there's white spots on here, and we can focus on what these little white spots are in contrast to the blue. This is a morpho also. It lives in the same place, but it's snow white. So my question back to Shu, and I haven't pushed this one at her yet, this is the first time she's seen this. What does it take to flip this thing here to doing that? Is that one gene? Is that just a little change in architecture? Or is there something much more complicated? In other words, evolutionarily, when I move from this to this, is that really a trivial movement? Or does this thing have to get from there to there? We're looking at 10 million years of very, very intense selection for some very different structural pattern in the wings and so on. That's a very real question to a biologist. That's a very real question. I have absolutely no idea. I've been looking at these things for 45 years. No, I correct myself, 60 years. And it never occurred to me to ask that question, even though I know these two are closely related to each other. Okay? So that's the kind of thing that, and Tresch asked me, English asked me, how do you connect to Shu? I don't in the beginning until I realize that there's somebody who can begin to address those kinds of questions. I can't, and I never will. I am never going to build a laboratory that can do the stuff she can do. What I can do is get the raw material for her to play with. Here's what that butterfly looks like. This thing is this is the size of a sheet of paper. It's that big. Okay? I've only been able to get a photograph once in my life of it. There it is. You have to strain and think a little bit. But see that white spot right there? One wing here and the other wing there. He's 20 meters off the ground. They live up there like big sheets of white paper flying up there. And in fact, the caterpillars, which is what I look at, are down here. But the butterflies are up there. So what you know is that at some point, mom says, I got an egg ready to go. Lays an egg and back up again 1 minute later. So she leaves a footprint down there called a caterpillar, but she lives up there with this enormous white surface. Now, what are the white saying? Why do we have these things that look like this? They're really obvious, these butterflies. Why aren't they hiding? Why aren't they cryptic? What keeps them in the game? What we infer, and there's a little bit of actual real evidence for this, is I'm baby bird. I grow up. I catch insects. I eat insects. I see this butterfly, bright blue, very obvious. Or I see this, bright white, very obvious. And me, teenager, I go right after it. And I never catch one. After the 5th one or the 10th one, to hell with it. And from the rest of life, this guy's got immune protection from that bird. Because what it is, and you learn this as a kid, I did, as a butterfly, collecting butterflies in Mexico, there's only one way you catch one of these guys, is to come up from underneath it like this from behind. It's the blind spot on a fighter plane that the pilot can't see in, and the butterfly can't see your net coming. And that's the one place a bird can come from, and there is one bird who specializes in catching these, nobody else does. It's called a jacamar, and that is exactly where the jacamar comes to catch those butterflies out of there, from behind like that with its bill. That's what those bright colors are for. Now, Shu showed this picture, and of course, what I come back to immediately is, it's striking that you think of these as being in the Mediterranean or in the tropics in Florida, someplace like that, Brazil. Why? Because in the wintertime, you want your roof to absorb heat. It's in the summertime that you want to reflect it. So now I put a challenge back to Shu when she talks about a white roof like this. Can you make a roof that'll turn black in the winter and white in the summer? Now let's look a little more at these guys. Now you know what this butterfly is. You've seen it with the blue marks on it. Let's focus on the underside. It's sitting here feeding on some fruit in front of our house in Costa Rica. It's a piece of damage to the wing, so you can see the blue. But don't think about the blue anymore. I should add, incidentally, when you were looking at these photographs, I suspect almost everybody in the room looked at this and looked at that and didn't look, that's just a big brown butterfly. Well, actually, the real interesting story, the other interesting story, is on this underside. So here's our butterfly sitting on the ground feeding on fruit. There. Now, everybody look very intently at the screen. If I had you wired in a psychology class so that we knew exactly where your eye went the minute that picture went on the screen, everybody in the room put their eye right there. Out of focus, not sharp, blurry, indistinct. And everybody in the room, eye went right to there. You may not think yours did. And you may then focus, oh, no, this is all blurry. So now I'll look at this. What's he got on the screen? Where's sharp here? Must be something in the photograph. If your eye didn't do that, you're dead. If you didn't react to that spot with focused attention, you were lunch 6,000, 6 million, 10 million years ago. Because this is what it really looks like. That's the eye. So you say, well, what's important about an eye? Like I said, if you don't notice an eye, you're dead. Because an eye means a predator. And I mean that very, very literally. You don't think about it, but you were just literally lunch for saber-toothed cats. Okay? Because if you don't notice that eye and pay attention to it, you end up looking like this. Now, the you in that sentence, of course, is every little bird in that forest. So that's what selects for that very fancy eye pattern. That's why those rings are on the underside. And Shu showed that picture that had the eye spots of the satyrid butterfly sitting on the ground with the little white dots in the center of each one of them. That's eye shine off the eye, of course. So you're the little bird. You come around the corner, and there's something that resembles an eye. If your reaction isn't gone, you're dead. If you stop to think, is that a frog? Is that a snake? Is that a caterpillar with an eye spot? Is that a butterfly with an eye spot? One time it's going to be a snake, and you're dead. And that little bird wants to live 20 years. Okay? Now, there's another aspect to this. You see, what Shu has done to me is made me start looking at more things than I usually look at. So here's our morpho again. See the blue up there from that damaged wing? Here's our morpho on the eye spots on the underside. And you say, oops, wait a minute. Look at all the eyes. No snake has all those eyes. That bird and you are hardwired to react to an eye. I don't care whether it's 1 or 5 or 15. Okay? It's doing something else. This is a rotten banana. And there's a tongue stuck down in the rotten banana right there. Let's look at it more closely. Now here's the head, there's the eye, and there's the tongue going down here into the banana and sucking up juices. Now think about this for a moment. This butterfly is standing on a rotten banana and sticking his tongue down into this gooey, sticky mess. It's going to pull his tongue out. And the tongue is absolutely clean. No bacteria living in it, no fungi in it, nothing glued to it. Think about it for a moment. Just a short time ago in the New York Times, or I think it was the New York Times, was an article about the molecular people who want to develop effectively a straw that they can stick into a single cell and suck out a single chromosome or a single piece of a chromosome, i.e., in their terminology, a single gene. And they're looking for ways to make a straw that can fit down into that cell, go all through all the blood, all the tissue, into that cell, pull this stuff out, and not be contaminated, not get jammed up, not get clogged up, and suck up the one thing they want to suck up. So my immediate thought was when I thought, hey, these guys have been doing that for millions of years. Now, that's not as thin as Shu will want to make it. But this is a big butterfly. You take a little tiny moth that's got a centimeter across its wings, it'll have a tongue the diameter of a human hair, which is doing the same thing. Okay? And then you start looking around and thinking, whoa, and if I'm thinking about that, look at these ants. They're walking right in the soup. Their feet are absolutely clean. That ant walks out on a piece of paper and there's no track, there's no bits of banana stuck anywhere. So what I want you to do is that when you see something like this, instead of just thinking pretty, color, I want you to start taking whatever you have with you at that time in your head and on the circumstances to look at it and try to say, wait a minute, why has it got those colors? What does those colors mean, that pattern mean to it? This is actually a moth, not a butterfly. So I want to close again with a slide I started with and just say to you, these are my clients. What I'm talking about in my terminology is ivory tower science on the one side, but it's biodiversity development on the other side. And why I do it is because if we can get these guys integrated into Shu and the rest of society, we have some chance that this will be here with us. Thank you. I have to explain that because I am essentially deaf, for questions, Winnie has to hear what you say. She tells me, and then I have maybe some chance of answering. I can see from all the gray hair in the audience that some of you are perhaps in the same circumstance that I am.
So we'll all be talking loud. And we want to make sure you get some questions. I'll start with just one quick question to Shu and Dan. Dan made this interesting point that he asks, when he looks at the colors, why are the colors this way? And Shu asks, how are the colors this way? How do they get to be this way? And I'm wondering whether you have started to ask some why questions, Shu, like thinking about what the kind of selective pressures are that produce the kinds of structures that you're studying and replicating. If that is useful to you in doing engineering and thinking about the structures and possibilities you might be able to make. And whether, Dan, you've been thinking new kinds of how questions at a more fundamental level, at a more kind of structural level than simply the visible morphology. So for both of you.
Go ahead.
All right. So as I mentioned, the very beginning reason we're interested in this kind of color is because interested in the photons, right? So we're interested in how we guide the photons, how we guide the light, how we utilize for technological application, and either is reflecting the light, scattering the light, or guiding the light. So to guide the light, all the light guiding is depending on the structure. So that's our initial interest, is whether we can make some kind of structures in either fancy way or cheap way. And of course, nowadays we want to make them cheaper, so make them very large scale. So initially is to understand the structure-property relationship, optical property versus a structure. And then, of course, we add the spin is to be mimicking the butterfly. And that's why I think brought Dan and I come together. And what Dan shows us, there's something much more interesting rather than just looking at technology, is how you impact the human, how you impact the society, how you impact the ecology. And this comes back to us. Now we actually can access the real sample, the raw sample, and the raw observation from Dan. And then the question is, what is the evolution question? Why do they have those kind of structures? What is the advantage having those structures? How the nature is actually fabricating those structures. We want to understand how to do this. So that's how we started to have those questions. And then I have very close collaborators. One is Randy Kaimya in physics, who studied the geometry. And the other one is Keith Stevie in the chemical engineering to look at the mechanisms, the interactions. So we want to put we have a team to want to put together is to understand. So first of all is whether we can reproduce the structure in nature, which is of course very, very difficult because they have many years of evolution to make those structures. The question is, how do we actually reproduce them, right? To reproduce them, we want to know why they have those function, why they have those structures. And if we can understand the mechanism, then we can actually maybe find another way to produce them much easier. We don't have to wait for many years of evolution to produce those interesting structures. That's our goal.
Great, thanks.
For me to address this question, I need to explain something. I spent 20 years of my life looking at why plants have got this and that chemical in them. So my question would have been, why has coffee got caffeine in it? Well, the simple answer is caffeine is the armor against all the guys out there who would like to eat that coffee seed. Okay, that's why caffeine is in coffee. It's not to make you happy. Now, my colleagues throughout ecology who ask those kinds of questions, traditionally, when you came up with that kind of question to ask, would turn themselves into laboratory chemists and build in their labs the technical capacity to find out how much caffeine there is in a seed, for example, how much tetrahydrocannabinol is in there in a kilo of marijuana. I went the other direction. I said, okay, I want to know how much is there in this seed and that seed and the other thing. So I would say to Shu, I want to know why and how, but I'm not going to try to bill it. I look around for the best people who do that, and I'm a parasite on them. And so what I say is, I'll get you a kilo of this stuff if you'll find out for me what's in it. And then we'll feed it to white rats or to people or whatever. And then we'll see what the consequences are. And we'll go back and forth. But I won't even think about doing what Shu does. I would not dream of trying to mount the army of postdocs and undergraduates and laboratories and the building and all the other stuff that Shu manages to make these nice pictures and nice understanding happen. So what I do is grovel around in the mud and find the organism, see what it's doing, and then pass the story to somebody else.
But yes, it stokes our curiosity, certainly. We think about it, but you know, we only think about as much as we can think about before we pass the material on to Shu.
Because I can't see what she can see. I don't have the machinery to see it. And we're looking at the genetics of these things too. I can't do that stuff. Oh, our biology department's full of people who do it all the time. But I find somebody in the world who does it that way very quickly, the way I want to do it, and I then provide them with the raw material, and then they get their thing out of doing it.
Yeah, what I found is interesting is because initially we're just looking at the structure, looking in the laboratory structures, we were designing the structures, but then there are so many other interesting things already developing in nature, right? We don't have access to those samples, we just think in the lab. But now we actually have something to inspire our thinking and to think about whether what's already done in the nature, whether we can reproduce and re-engineer them, right? Inverse engineer. So we know there's this kind of functionality. Can we actually re-engineer those things? So I think that's why it's interesting.
It's also interesting just because it seems to be a case of mutualism, of coevolution. Like you're imagining your ecological niche.
Deliberately,
These are the things you can do, these are the things you can see. In order to attain these other ends, you have to bring somebody in from another niche who has their own specialty. So it's an ecological vision of the disciplines that's getting remade with these kinds of collaborations. Questions from the audience? Yes?
I thought that was very interesting. Just wait for the microphone, we'll bring the mic up. Scanning electron microscopy pictures in terms of the way in which structures are built that are responsible for some of the output that you get from that particular structure. Are you using in your work something like the scanning electron microscopy aspects of a structure to be able to use that as input to a 3D printer where you can actually produce these different types of structural analogs that might have different physical properties associated with them to give you enhanced or even the same properties that you're trying to get?
Yeah, so absolutely. It's a very good question. Initially we just designed 2D structures, but now we see in butterfly wings, there's so many, it's a 3D or hierarchical structures. It's very difficult to fabricate them in the laboratory. So nowadays we have more advanced technology called 3D printing. Right, so we're actually very interested to look at using the scanning microscopy or using other type of techniques to look at the 3D imaging, and then whether we can reproduce them. So there's different landscape questions. There's question of how do you advance your technology to actually not only make micron size, maybe even go to smaller features. So that's a lot of challenge of this. So we are interested in acquiring 3D printer and then optimize this and to make to write the smaller features. So tomorrow, I'm going to talk to another Xerox retiree to do inkjet printing. All right.
But one interesting thing I saw in your text is that that's not the only way you have of reproducing the micro scale. You have a whole set of different kinds of assembling techniques, including different polymers or different solutions that will sort of brutally transform, in the case of the butterfly wing, it will not be a smooth lacquer, but will alter make a rough finish, which makes it hydrophobic. So it's by pouring a kind of solution on it that you alter the molecular structure. It sounds like you have many, many different strategies for assembling these kinds of compounds.
Yeah, so 3D printing is sort of like top-down process, right? You know the structure, you write the structure, but also as a engineer or material scientist, we have other tools called self-assembly or bottom-up approach. So I can manipulate the material. So I put a sort of skeleton in the 3D using the 3D printer. But then I can manipulate the chemistry or the mechanical properties, how they swell, how they interact with solvent. I can manipulate how the pattern is actually changed. So you saw in the ice spot Dan was talking about, there's a lot of this kind of wing patterns. It's all because of their so-called, like how you form wrinkles. So you can play with hardness, softness to manipulate how the pattern is altered within the 3D skeleton. That's our interest.
Yes, microphone.
So I'm curious to know, how much do you think about the material itself that you're using? Do you look at biodegradability of the material that you're using, or do you think in the future, when someone gets to the point of manufacturing, do you look at the effects of manufacturing so it doesn't actually destroy the things that it references?
Oh, absolutely, yeah, so absolutely. So depending on the purpose, right, so we can either make them very, very stiff, like metal, or very strong, or we can make them biodegradable. So those are different aspects we are looking at, so it's all manipulating materials properties. And so the other aspect I think is very interesting to me is butterfly, for example, morpho is huge. They're very agile, but they actually, it's very lightweight, but very, very strong. So that's another aspect I'm actually very interested in this, other than color, is mechanical properties.
So to put that to Dan, I mean, what are your feelings about the ecological footprint of nanotechnology? Is that an issue for you? Is it a question you ask before you enter into a collaboration like this?
No, for me, the ecological footprint of nanotechnology is in no qualitative way different than all the other footprints that we leave out there. Okay, so we leave a lot of different footprints, and whether they're through nanotechnology or whether they're through organic chemistry or whether they're through transportation and highways, it doesn't matter. All of them are impacts. I personally am of the school that says, take a deep breath here. The only real biodiversity that counts, certainly in the tropics where we have a lot of it, that's going to survive is a few big patches that we come to sort of a peace treaty about. That society will in fact leave them alone because they're useful in a variety of different ways to a lot of different sectors of society. And frankly, in my mind, the bulk of the rest is gone. Whether it's gone in 2 years or 5 years or 15 years, it's gone. Think Iowa. How much original forest is there left in Iowa? None. Indiana, almost none. That same thing is happening throughout the tropics. We just did it a little earlier in some places. So the only pieces that are going to really survive of significant blocks about all the butterflies and the birds and the beetles and the whole package are where those societies that have those blocks come to some kind of a peace treaty and say, okay, 500,000 hectares over here, that's just as valuable as 500,000 hectares of cornfield over here. So our society will have two kinds of crops, one the wild crop and one the classical agricultural crop. Those are the only ones. So that the footprint inside, we basically minimize it. The footprint outside, we forget about. Just like I forget about cornfields. I don't try to save what's in Iowa cornfields. Okay, that battle would have been fought 200 years ago. It's gone now.
Interesting.
But I would add, frankly, we don't know enough. The footprint, some of the modern technologies are actually very pervasive, so you can't really wall yourself off from them by distance. And we don't know enough. Whatever the footprint is of nanotechnology is showing up more, I think, in aquatic systems right now. Surely it's affecting soils, but we simply don't know. We do know about some chemical effects of herbicides and pesticides, and from the little that we do know, they are very bad. But there are so many areas that we really don't understand at the level of our clients.
Okay.
Probably the most dangerous one I can imagine quickly off are the self-replicating robots.
Sure. Yeah, they're bad. We can agree on that. Other questions?
Hello. Thank you for that talk. That was great. That was a great talk. Starting off with that slide that you had, that you talked about your clients being like, you know, like the inhabitants of that of the reservation. I was just wondering if you would like if you envision yourself telling like their full story, also from a cultural perspective, maybe bringing in like maybe folklore or something else that gives like a more broader picture. So I was wondering if in your team you have you envision a team of just strictly hardcore scientists, or you also want to have like historians or cultural, you know, like someone who deals with cultural diversity or whatever, like from a historical point of view. Yeah.
You can speak up. Go ahead.
I'm going to be a little loose with the question, at least as I'm thinking of passing it on to you, is whether our team includes people who are not hardcore scientists, but are also involved with people, and particularly in our cases with historians, not so much with us, with folklore, things like that. I'm thinking Javier Basurto. I'm thinking the role of the paradoxonomists in their neighboring village. I'm thinking that the very people that you're thinking of are paradoxonomists doing hardcore science, are actual living examples of people who are holding parties in their schools, who are competing with other parents in the science fair, who are just telling their neighbors what they're doing. And I will now pass the question on to you.
Well, no, I'll just add to what Winnie just said, two things. One is that we don't make a point of looking for the ivory tower to come and study what we're doing. When they show up, they're very welcome. We have one very nice Mexican bicultural U.S.-Mexico graduate student, finished, got his PhD, he's now a professor at Duke University, who did his entire PhD studying the paradoxonomists, how they think, what they do, why they do it, and living with them for 2 years. But that was of his own volition. He came to us to study us. The other side of the coin is that, just as a concrete example, 3 days ago, I got an email from one of the paradoxonomists that the computer was down. When he answered it back to take the laptop to another paradoxonomist and start working off a notebook again until we got a chance to get down there. The teenage son of another paradoxonomist hits Winnie back at 10 o'clock that night saying that he's run diagnostics on the computer, figured out what's wrong, and here we go. Now, his parents did not make it through grade school. Okay, so that's our interaction, is out laterally through the community rather than studying it up into the academic community, which then maybe gets into policy, which maybe gets down somewhere later on. So what we're trying to be is a virus working at this level. Now, and I say that very literally, I didn't understand it in 1985 when we started this. We found ourselves, after a short time, being accused of what you would call, a historian would call, a peasant revolution. Because when you take people who are cutting cane, herding cattle, riding fence, washing dishes, babysitting, whatever it is, and give them the tools to do the kind of stuff we're talking about here, that is not welcome by the middle nor upper class. Because you're creating competition for them, and they don't want the competition. And that's the world that we live in. So I just add that to the question.
But I'd follow that with the question with a thought that perhaps bringing in that ground level knowledge of the traditions and the culture may be useful for building these kinds of peace treaties you see as necessary for preserving these large spaces of biodiversity.
Yes, if you don't have people managing it, it's finders keepers. That's the lesson we learned in 1985. To put it in just a couple of sentences, we were broken out of the ivory tower by an incident where 1,500 gold miners invaded a national park. And 1,500 gold miners can trash a national park really well. And in talking to the gold miners who were accused of every sin on the planet, what we realized suddenly was they felt morally perfectly justified because it was just no man's land. So what? Why not? And furthermore, we're helping to solve the national debt.
Right.
The central bank opened an office in the national park to buy the gold. Okay? Now, we saw that and we suddenly realized that, whoa, the only way this wild area is going to survive is if it's viewed as a productive part of the rest of society.
Right.
So that means we've got to treat it like a farm. Occupied, doing, producing. That means some footprints. To go back to the nanotechnology question. That means there is some road, there is some buildings, there are people, there are things doing things. But you hold it down to a low level.
So I think we have time for one last question.
To follow up on what you just said about wild areas having to be viewed as a productive part of society, do you see this cross-fertilization between biomimicry and engineering new technologies based off of nature as playing into that equation? That is, do you see nature as having intrinsic value for innovators and engineers, and does that play into the conservation
Absolutely.
debate?
Absolutely. When I say biodevelopment, I mean anything. If Shu takes that butterfly wing and turns it into a product that matters, I grab that and I put that in the face of society and say, this is a product of a wild area. Sure, it's a product of the ivory tower. It's a product of engineers. But the raw material is coming right out of this place, and if you didn't have that raw material coming out of that place, you wouldn't have that product. And so you'll find me in there grubbing and just like an agent for a Hollywood star. I want a royalty. I want advertisement. I want acknowledgement. Not super greedy, but the same as any other business person would. But not for me, but for the budget of that wild area.
Just a last thought for Shu. Have you gone green as a result of your collaboration with Dan, or is that still down the road?
So that's why I think it's very interesting, because as I mentioned earlier, so the biology is much more complex than what we produce in the laboratory. So there's two routes, right? So one is you perfectly replicate whatever is done in nature and using the 3D printers or some advanced technology. And the other is to understand the mechanism, to understand how they process, how they produce those structures, and then we can re-engineer our mechanism, our materials, to actually generate something more interesting or even better. And more interesting to us is whether we can manufacture this in large scale. So that's very, very challenging to us, but on the other hand, I think there's a high reward. So whether we can actually build it's not just the mimicry, it's actually make the butterfly buildings, right? And then we can have this everything cheaply and impact how the people think about the environment. The environment is not just the environment, the wild environment, it's the environment in this building, how you should behave. I think that's what we are interested in, to contribute to that.
I'll take that as a yes. So thanks very much, Shu. Thanks, Dan. Thanks, everyone.